Hello everyone, welcome back to another class on regeneration biology, and today we will learn about organ regeneration in an overview. We will dissect one by one. Organ regeneration, by now you have a clear idea that we have seen different types of tissue regeneration, and organ regeneration in simplistic sense means restoring a damaged appendage or damaged organ in toto in the place of a missing part. So that is the simplistic sense, is the organ regeneration. And I'm sure all of you are familiar with this animal picture. This is axolotl. It's also Americans call it as axolot. L is silent. In either case, this is one of the fantastic animal used in regeneration biology studies, and it is native of Mexico, seen only in one lake, Shoki or something. You can Google it, you can find it out. Only in one lake it is been seen. The most important thing is, although we have plenty of relatives of axolotl across different parts of the world, axolotl is seen only in one place, and that too restricted to one lake, not even to one country, just restricted to one lake naturally. But it is there all over the world. In nature, it is almost extinct. It is not there at all in the nature in Mexico, and its scientific name is Blastema mexicanum, as you can see here, and it has been extensively grown in laboratory and also in pet shops. People use it as a pet animal. And interesting thing about this animal is it has got gills, and it also has got lungs, and it can breathe air as well as in water. In other words, this animal shows a situation called neoteny. So neoteny basically means the larval stage. So this, what you are seeing in this picture, is an adult, but it did not change from its larval stage at all, except that it increased in size. It retains every larval characteristics. You may wonder what larvae you are talking about. It simply means every salamander has got this larval stage, but it metamorphoses into adult, and which is a land dwelling form, and it can breed, come back to water for breeding purpose, because the larvae is again in aquatic. But axolotl choose not to metamorphose by default. We will not get into the details why it doesn't metamorphose, etc. If necessary, that time we will discuss. But understand, by default, the larvae becomes adult without undergoing metamorphosis is called a neoteny. And this probably could be one of the main reasons for its remarkable organ regeneration capacity, because every animal, including other frog species or other salamander species, the larval stage has got exceptional regenerative capacity. Even for mammals, mammalian, or even chicken, or any other non-regenerative species, also in embryonic stage, they have excellent regenerative capacity. So can it hold the clue that neoteny feature is allowing axolotl to regenerate efficiently than any other animal? We have to explore it. So this is one old issue of Science, I think 2005. So in that, they have discussed about different questions. What it says, 125 questions, what don't we know? So it's basically addressing that what are the challenges that the science has, and what are the challenges which we should be exploring for making a better human life. So in that, they talk about one interesting topic, that is what is mentioned. Those who are interested can read it. I am not going to read it out. One page has been put. What controls organ regeneration? That is one of the 125 questions. What controls organ regeneration? That means every animal has the capacity to regenerate a damaged organ. Like we regenerate liver exceptionally well, we can regenerate a nail or a hair or skin, etc. We do regenerate a lot of tissues in our body, Blood cells we constantly regenerate. Your RBC lifespan is just 4 months. WBC is only around 90 days. So we regenerate constantly. We regenerate these blood cells, and they die also. Otherwise, every human will live only 4 months, because if the blood you are not able to regenerate. So we do regenerate, but when it comes to complex tissues, we lag behind or we do not regenerate. So organ regeneration is one classic field of study. If your kidney is damaged, why we can't have a kidney regeneration? But when liver is damaged, liver can regenerate. So intestine, if your intestine epithelium is damaged, it can regenerate. But if your intestine is chopped off, new intestine will not form. So these are all some questions which baffles scientists. So it is widespread phenomenon in a group of animals. You can see hydra here, planarian here, frog and earthworm, fish, mouse, various crustaceans, Drosophila, and salamander such as axolotl, and also human hand has been given. Human hand, we are referring to the bones basically. If hand is chopped off, new hand will not form. But if a bone is fractured, it heals. Not perfectly, but it heals so that the continuity of the bone is restored. So all these species are blessed with the ability to regenerate, and they are extensively used as a model organism. So there is variability in the ways that the animals regenerate, suggesting that the regeneration has evolved multiple times in evolution. Therefore, there are likelihood that there will be multiple strategies to consider, and each model system has something to teach us how they are doing the regeneration. This view begs the question of why humans have not evolved the ability to regenerate. There are so many there are so many reasons and arguments can be there. There is also one theory, when a group of humans that are migrated out of Africa, that probably you already know it, that a Indian and an American or a Japanese or a Chinese or any other ethnicity, they are more similar than a person living in Africa, in one village to the neighboring village. The distance between those two villages may be only 10 kilometers. They are more diverse than you to American or you to a Chinese or a Japanese. So this may sound baffling. So the reason being, there are multiple rounds of migration has happened out of Africa, but in evolutionary timescale, it has happened that in some stage, the human population outside Africa, remember, outside Africa has come down to as small as around 1200 people. Around 1200 people have come down, and they colonized the rest of the population. So 1200 is a small cohort considering the human as a species. But what you get because of this, whatever feature and qualities those 1200 people had was enough to make them fit and survive in that environment. That may not have had this so-called feature of regeneration. Hence, the rest of the population did not have. This is one theory you can put forward. What do we need? Why and how and what we need to regenerate an organ like a limb? What are the stuff needed? It's just like asking, okay, I need a hand to be regenerated. What are the, like people say, your hand, your palm is the second most complex organ after your brain. I think it has got around 27 different bone pieces, just your palm. 27 bones, around 30 different muscles, and around 3 unique nerves that controls, and 3 or 2 nerves, and major arteries, 2 or 3 arteries, and 2 or 3 veins. These are all unique. So 30 muscles, different muscles that has to be controlled by your brain is a complex task. So what I mean to say is that your hand regeneration is not just a structural regeneration. Its a function is very complex. So although your fingertip is a simple organ, when it comes to regeneration, it is not just make a copy of hand. It is making a functional hand. So that involves a lot of complex trajectories that has to come into picture. Regeneration competent cells that can do what need to be done. They have to migrate, they have to proliferate, and they have to differentiate. So this is very, very important. And the regeneration permissive environment, that is the regeneration permissive niche, is a must. If the regeneration do not happen properly, if the regeneration do not work effectively, then what can happen is it can influence the survival or the selection of that cell on that spot, can get negatively affected. So even though ability is there, like you are a great singer, but I have put a tape on your mouth, so how are you going to sing? So same logic, I have a singer doesn't mean that song is a given or song is a fact. So regeneration instructive environment is a must. If the regeneration is not possible in a given environment, the capability to regenerate is not going to work out, although you have the capability. So regeneration is the reactivation of development in post-embryonic life to restore the missing damaged tissue. So during embryonic development, like in around 2 months of gestation, your hands, legs, everything is formed. Your size may be very small, maybe size of maybe 2 or 3 centimeter, but you have become a full-fledged organism. Even the hands, limbs, I think around 3 months or 2 to 3 months of gestation, even your fingerprints are formed. You can imagine the complexity of development. Your thumb or all individual fingerprints are formed around that time of development. But can we recapitulate it? If an organism is 20 years old, can we recapitulate? Can you reactivate that 20-year-old off pathway? That means if you are 6 months as an embryo, say, or a newly born embryo, that is around 9 months of development, but it has stopped limb formation around 7 months ago. It is not having, limb is growing, but no embryo is developing new limb. So if you are 20 years old, you can imagine 2 months into gestation, the limb formation is stopped. It is a very unhuman phenomenon that led humans to wonder why we cannot regrow our arms and legs. It is unhuman phenomenon. What it basically means, we all want it, but we are not capable of. That means it makes a person debilitated. What gives other animals the ability to regenerate tissues while we cannot? So these are all some interesting questions that you can see in that science article that is published. So regeneration can occur in 4 major ways. We have seen them one by one. We are recapping that. Stem cell mediated regeneration. Stem cells are the sources of regenerating the lost tissue or organ. Example, hair shaft regrowth from stem cells in the hair bulge and the replacement of blood cells from the hematopoietic cells. So we know these are all formed because of presence of unique cells. We can call them as adult stem cells. Another thing is epimorphosis. Adult cells in some species can dedifferentiate into an undifferentiated mass, which then redifferentiate to form new structures, for example, in regenerating amphibian limbs. So there is no stem cells available. They are formed based on demand in that spot by the dedifferentiation of the existing tissue. Then comes morphallaxis. Morphallaxis also we have seen it. What does it mean? Regeneration occurs through repatterning of existing tissue and there is little new growth. Example, regeneration in hydra. Say you took 100 cells or 200 cells from hydra, just a cell assembled in a petri dish. So it immediately forms a hydra, miniature hydra. Or you took a normal hydra, chopped off its head. So it has got only bottom portion. From the bottom portion, it will use the existing cell. Say a hydra had 1000 cells, let us assume. Chopped off from the top, 500 cells are gone. Actually, 1000 cells are there in a hydra. You assume it's not a fixed number. I'm giving for an example. 500 cells are removed. So the remaining 500 cells, both the pieces, the top piece and the bottom piece, they rearrange and form a normal hydra, miniature hydra. So repurposing. So some of the base cells or the stalk cells now assuming the tentacles, head, etc. Same way, the head portion now have to repurpose the cells into the stalk and the its basal foot, etc. It has to repurpose. Then it will increase in size. So this is morphal axis. And then comes the compensatory regeneration. Here, the differentiated cells, say liver regeneration, divide, but maintain their differentiated function. That is, they observe normally seen in the mammalian liver. Simple example, I have 5 fingers, 2 of them are cut, and now these 3 fingers are growing and occupying this space. Only 3. This becomes double the thickness, this becomes double the thickness, this becomes double the thickness, so that it occupied these 2 areas. Or if I chopped off all the 4 fingers, this thumb is now becoming like a paddle, becoming big, so that it acts like it. So that is compensatory regeneration. So if you look into the epimorphic regeneration in salamander limbs, normally it takes around 2.5 months time it takes for complete regeneration. On an average, you can tell 60-65 days, and this is up to 72 days. Picture is been shown. So 2 things are shown in this. One is distal amputation, another is proximal amputation. Proximal amputation means you are amputating close to the body. Say for example, you are cutting here. That is proximal. Cutting here is distal. Here is proximal. So proximal closer to the body, distal means away from the body. But no matter where you cut it, by around 72 days, both have grown. So not that if you cut here, it takes longer days. If you cut here, it takes only shorter days. So what you understand, if the damage is here, the growth rate will be faster. Or if damage here, the growth rate will be slower. That is what you are seeing here. If you cut here, it is growing at a rapid rate. Whereas you cut here, it is growing in a slower rate. So understand, no matter where you cut it, the time taken is going to be fixed. When a salamander limb is amputated, the remaining cells regenerate. That is, once you remove these cells, what you are talking to, or these cells we are talking to, remaining cells, and they will de-differentiate and they regenerate a new limb, which is a complete copy of or mirror image of the other limb on the other side, with all differentiated cells arranged in proper order, a functional limb. Remarkably, the limb regenerates only the missing structure and no more. It cannot generate other than any extra. One limb is cut, 2 limbs are not formed. In some ways, the salamander limb knows the point to the proximo-distal axis. This is what proximo-distal axis, where it has been severed or amputated, and regenerates from that point onwards. If it is cut here, it will not grow from here. If it is cut here, it is not growing from here. Not that here I will get a palm formed. It will make the entire thing. So the salamander accomplishes epimorphic regeneration by cell de-differentiation to form regeneration blastema. We have seen this terminology multiple times, and which is an aggregation of relatively undifferentiated cells derived from the originally differentiated tissue, which then proliferates and redifferentiates into the new limb structures. So you can see here in its ultrastructure, the source of regeneration competent cells, how it's being formed. The regeneration blastema is equivalent to a developing limb bud. The apical epithelial cap, also known as AEC, is equivalent of apical ectodermal ridge of the developmental stage, where the limb is formed. Actual limb is formed is from AER. Sonic hedgehog signaling comes into picture. That decides how you should have different shaped fingers, etc. I will not go into the details of that. But AER is a major source of, it also uses hedgehog signaling. So there is a blastema mesenchyme. So this is a limb and this is a blastema picture, and this is called a mesenchymal cell. Epithelial mesenchymal transition we discussed multiple times, and blastema mesenchyme, and it's equivalent to the limb bud mesenchyme. And then here is the zone of dedifferentiation is unique to regeneration. The zone of dedifferentiation is unique to regeneration. And then there is also a mature differentiated proximal tissue. There is a mature differentiated proximal tissue that are source of the blastema. So there is a differentiated tissue which give rise to the blastema cells, and they give rise to a cap. So we can also see the dermal fibroblast, they migrate and proliferate to form an early regeneration blastema. So you have created an amputation here, and it forms a small lump, and then it increases in number, and up to 80%, that is nearly 50% on an average, of the early blastema cells come from the dermal fibroblast. So epidermis is the skin, and below is the dermis. You can see here, this is epidermis, this is dermis, and this dermal fibroblast and epidermal is forming a cap. Apical ectodermal cap is being formed, and these dermal cells, they migrate into the wound site, and they are the actual source. People have shown this by staining approach. Now also you can see this red color cells, there are plenty. Although other cells also come from here, because there will be also fibroblast in there. But the major source of around 80% on an average of the early blastema cells have to come from the dermal fibroblast. So the formation of the apical ectodermal cap and the regeneration blastema is been seen here. So you can see this is a humerus bone, and this is the epidermis, and this humerus bone also need to be formed, you should understand. And when you block it, there was an amputation site here, and the blastema has formed, and then there is a here also there is a muscle cells that is developing, and epidermis and the humerus bone grow hand in hand, and the apical ectodermal cap, they remain intact. Even when the blastema is growing further, the apical ectodermal cap also is growing forward. And then you have the blastema here itself. It is their blastema, which is in the extreme tip of it. And then you can see here, these mesenchymal cells, they migrate, and the blastema become mature and mature and mature. Then one more thing you should know, near this bone, what is present, they continue to differentiate. Or in other words, this zone is the place where the active differentiation is going on, and this zone is the place there is active proliferation is going on. So here, although proliferation is occurring towards the proximal side, it is more of differentiation, because this blastema continue to grow further and further. So when salamander limb is amputated, a plasma clot forms, which has got fibrinogen, etc., which allow these cells to migrate easily, fibronectin. So within 6 to 12 hours, epidermal cells from the remaining stump migrate to cover the wound surface, forming a wound epidermis. It forms a wound epidermis. Below that, the blastema forms. In contrast to wound healing in mammals, no scar forms, and in dermis, it does not move with the epidermis to cover the wound. So wound healing is seen in mammals, but in this animal where the regeneration occurs, scar is not formed. In fact, scar is formed and the scar is removed instantaneously, and so that the regeneration is kick-started. No scar forms, and the dermis does not move with the epidermis to cover the wound. So this is what normally you see in the case of a typical wound healing. If the dermis moves, then there is a chance it can give more of the blastema cells. 2 days after amputation, the skin and muscles have retracted from the bone surface, and at 5 days, a thin accumulation of blastema cells is seen under the apical ectodermal cap, and at 7 days, large number of mitotically active blastema cells are seen distal to the humerus. So what you see here now is, in 4 days following the amputation, the extracellular matrices of the tissue that is lying beneath the wound epidermis are degraded by proteases liberating single cells. So the proteases allow cells to become free from each other. That means it becomes more of a mesenchyme and undergo dramatic dedifferentiation. Soon after cut, proteases comes into picture, single cells are formed and they reprogram and then start proliferating. Bone, muscle, cartilage cells and fibroblasts lose the differentiated cell characteristics. Differentiated cell markers are downregulated and increased expression of genes such as MSX1 associated with the proliferating progress zone seen in mesenchyme in the embryonic limb is observed. So MSX1 is a marker of blastema, and genes and increased expression of MSX1 is seen associated with the proliferating process that is nothing but blastema. And here also you can see they start becoming D1 and D2, digits start forming, okay, and different radius, ulna, and different parts, and this is humerus bone, all zones are there formed in after a few days. This cell mass is the regeneration blastema, and the cells will continue to proliferate and redifferentiate to form new limb structures. The wound epidermis thickens during this process to form the apical epidermal cap, which is similar to the AER during limb development. This is at 8-day blastema elongates by mitotic activity, and much dedifferentiation has occurred. And at day 9, early redifferentiation can be seen. A marks typical mesenchyme blastema as it is mentioned here, A, and H marks the proximal part of the regenerating humerus bone. U and R, they are cartilaginous structures of the radius and ulna, and P represents the stump where the amputation was done. This is the plane of amputation, and these are all the structure that is formed from the blastema. And at day 10, C marks the pre-cartilaginous condensation of the carpal bones ankle, and the D1 and D2 marks, that is the digits that is being found, D1 and D2 marks of the same for the first 2 digits, what you are seeing, D1 and D2, this one D2. So what are the contributing tissues of the blastema cells? Epidermis give rise to wound epithelium, which give rise to apical ectodermal cap. Stump cells give rise to blastema. And the contribution of blastema, if you look, total blastema you are taking, if there are 1000 cells in the blastema, you are looking. Dermis fibroblast around 19 to 43%, it can range. And muscle myofibers, actually in the stump there are 19% dermis was there, and it is now increasing to 43%. And muscle cells in myofibers 55% are there, that will reduce their quality and become 17% in the blastema. And in skeleton, the cartilaginous bone around 6% will become 2%. So in general, the dermis contributes a large chunk of the fibroblast. But remember, once they are all reprogrammed, they are all going to become typical normal cells. Tissues in the stump of all contribute to the blastema. Tissues in the stump do not contribute in proportion to their availability. Progenital cells are lineage restricted except for fibroblasts. They are lineage restricted when it comes to their fibroblast qualities, are even if it is there present, they do not have a they are not going to give rise to any tissue. Like in the sense, although the blastema is a mixed population, they retain some identity which they are going to give rise to, although they are undifferentiated in a common, but not that any cell can give rise to bone, not that any cell can give rise to muscle. So there are many challenges that are facing the regeneration. One is non-regenerating models are like playing in a lotto. That means if you can wound heal, you heal it, or if you get it, you get it. Sometimes you get it, especially if you are early stage of development. Amputation studies in the regenerating models are inherently loss of function analysis. So you study the loss of the limb, and amputations create noise, that is extraneous signals that obscure the regeneration signals. And many gain of function studies in the non-regenerating species is often incomplete. And this is a picture where amputated man, and there is a confusing road direction that is given to a cell that is trying to regenerate. So goal is to reduce the complexity of regeneration and design a gain of function experiments for the identification of the signals that are necessary and sufficient for regeneration. So this is the overall theme of regeneration, and we will study about regeneration biology more in the next class. Thank you.